Welcome back. We'll begin a look at the blood and see what we have going on in here. And so, back in anatomy, you looked at the blood as far as what was in it and its composition. And so you saw that in terms of when we spin blood down and look to see what's inside there, we see that we have almost half of it is plasma and almost half of it is red blood cells. And so the majority of the total component of blood is either plasma or red blood cells, with a very small portion of it being here in this buffy coat, where we have the rest of our white blood cells, the platelets, so the rest of our formed elements that we find within the blood itself. And in the lab, we would normally do a process to see what is inside there to determine the hematocrit of the blood itself. And in order to do that, we use a little centrifuge, and this centrifuge allows us to pick up some blood into these little tubes here, and then they get set into the centrifuge and spin it down. And so that allows us to then see what's inside it. So when we first collect the blood, we have essentially a combination of all the blood kind of mixed up. But what we want to end up with is a vial there where we have the blood inside there is divided up into its components. So it's going to be by weight. And so the heavier components are going to get spun down into the tube itself, which are going to be our red blood cells, and then our white blood cells, and then everything that's left in the plasma itself. And again, we're looking for something that is kind of mostly red blood cells, mostly plasma sitting there on the top. If we have a change in that, where we have either an increase in red blood cells or a decrease in red blood cells, we know that something's going on. And so something is happening inside the person there to either cause that increase in red cell production or in a decrease in red cell production compared to kind of where the normal itself is. And then it becomes a process of figuring out what exactly is going on within their blood themselves. All of those components that are within either the red blood cell portion there or the buffy coat are our formed cells. They all begin in the bone marrow, so they're going to be found within red bone marrow. Inside that red bone marrow, they're then going to be produced, and they're all going to start out from a common stem cell, which then gives rise to the different components of our white blood cells, our red blood cells, and then the platelets as well. And so we have different types of kind of beginning cells for those stem cells, but they're all going to begin from a common origin, a common source found within the bone marrow itself. So when we look at the different components within the blood, part of which are our formed elements, but the majority of which is actually plasma. Plasma is kind of the liquid portion that sits on top. So this is going to be almost entirely water. So if we look at just the plasma itself, it's almost all water. From there, we've got a large number of proteins. And so we have a large number of proteins that are in the blood there. Many different kinds of proteins, most of them all produced over in the liver. And they're going to help with many different functions within the body itself. And so they're going to be able to help out with everything from immunity, which we talked about a little while ago, in helping to give us that immune response to things within the body. So things like our complement and so on. Blood clotting, we're going to have different proteins that are in there for clotting itself on there. The ability to transport other substances. So sometimes things need to be kind of packaged in with a protein in order to be able to move them from one place to another. Things like hormones, non-water soluble substances are going to need to be able to kind of be packaged up as it goes along there. And then we get into kind of components where we're regulating things within the body, both water balance and pH balance, able to buffer the blood as pH changes at different times. And the same thing goes with the ability to balance out our water as well. And so within that, one of our biggest components of plasma proteins is albumin. And so that makes up a large percentage in terms of the types of different proteins that are there for them. And so we have lots of albumin inside there for things like Immunity, we have immunoglobulins, and so we have different substances that are there within them for those as well. From there, within the plasma, we then have kind of all the different things that our cells need. And so lots of different nutrients. And so things that we can move around within the blood itself. Things that are going to be needed to produce other cells, amino acids, fatty acids, things like that. We got all the gases that we transport, oxygen, carbon dioxide, fair amount of nitrogen inside there as well, moving around. And then we've got waste within the plasma as well. All the things that we need to get rid of out of the body, urea, uric acid, creatinine, all the different nitrogenous wastes that are produced by all of our cells all day long. The plasma itself is not kind of like a static thing. This is something that's going to change kind of throughout the day. At times, it's going to change moment to moment in certain substances. Other substances are going to kind of go up and down throughout the day, things like that, as different quantities of things come in. So the plasma itself, because it is almost all water, that allows us to have essentially all of the water that our cells are going to need at any given time. This allows for the filtration out of the blood and into the tissues of our nutrients, electrolytes that are able to be transferred over to the cells themselves. And at the same time, it's going to work as a solvent to absorb substances from those tissues. And so we can get our waste products, all of our nitrogenous waste moving back into the blood. We can get our carbon dioxide to come back in from the tissues as well. Because the plasma is almost entirely water, this also makes it a great way for us to alter the heat of the body. So if we're too warm, we can carry the heat of blood up to the surface and allow it to be taken away. If we need to conserve heat, we can absorb that through water as well and kind of get down deep closer into the core of the body as we may be getting cooler and cooler. Within a blood sample, we have different types of cells. We have things like red blood cells, we have platelets, we have white blood cells that are found in there, and then kind of all the empty space that is there is all the plasma. And so when we're looking at a slide with those different cells on it, those are the things that we can see. Lots of red blood cells, lots of plasma, and then kind of interspersed we have those components of the buffy coat, our white blood cells and our platelets. The red blood cells, when we look at them, they have a biconcave disc. So if we were to look at it from the side, they kind of dip in on the side there and then dip in on the side there. Almost looks like a dog bone or something when we see it there. And that allows for an increase in surface area. So large amount of surface area, lots of ability to get oxygen in and carbon dioxide out. And so we can change the components that are within those cells themselves because the inside of those red blood cells are almost entirely filled with hemoglobin. And so large quantities of hemoglobin inside there. So biconcave shape gives them more surface area. We've got better, quicker ability to transfer gases in and out of that cell itself. That biconcave disc as well is essentially created because it got rid of its nucleus and its organelles. So there's not really a whole lot inside there to kind of keep the cell inflated, so to speak. That's there. It's very flexible. It's got nice proteins inside its plasma membrane that are going to allow it to squeeze through those tight little capillaries. Remember when we talked about capillaries, we said that oftentimes the capillaries were actually smaller than a red blood cell, so they're going to have to squeeze and push through those tight, tiny, small areas. They're almost entirely filled with hemoglobin. Huge amounts of hemoglobin inside there. We'll talk about that in just a second. So vast quantities of hemoglobin, able to carry all the oxygen that it needs to. Red blood cells, unlike things like white blood cells, do last for a fair amount of time, upwards of 3 months or so. And so we have the anywhere between 3 and 4 months, so 100 to 120 days for the average red blood cell as it's out there doing its job. As it becomes more and more older, the flexibility of the plasma membrane becomes decreased, so it gets harder for it to squeeze through those small capillaries. It may become damaged along the way as well. And since it doesn't have any nucleus or organelles, it doesn't have a way to fix itself. So those cells are going to die off. And once they do, the spleen is generally going to be the place where we're going to recycle those cells. And then the spleen and macrophages there are going to break them down, and we're going to tear the hemoglobin is going to be our primary thing that we want to pull apart, and we're going to pull out the iron from there. We're going to pull out and break it down into globins and heme, and we can recycle these two, and the heme portion is going to become bilirubin. So we're going to have to recycle that as well, but in a slightly different way. So here we have our hemoglobin themselves. It is divided up into alpha and beta proteins. And so we have 2 of each, 2 alphas, 2 betas on there. We then have 4 iron. And then eventually once oxygen comes along, each one of those iron is going to bind, well, each one is going to bind 1 oxygen. So since we have 4 irons, we're going to end up having a total of 4 oxygen on each of those hemoglobin that we have there. The hemoglobin's shape will change just slightly as we see them there, as more and more oxygen comes on. And so each of the subunits themselves has the protein portion there, and then the iron. And each iron can then carry 1 oxygen molecule. So it has 1 oxygen molecule on each on there. Once it's dropped off its oxygen, it can also pick up carbon dioxide. And so it can pick up some carbon dioxide to carry back to the lungs. But the majority of that carbon dioxide is actually going to be transported as carbonic acid. And so we're going to move it through the system, convert it into carbonic acid, and then from the red blood cell, convert it into bicarbonate. And we have that outside of the red blood cell in the plasma to travel back to the lungs. In abnormal conditions, we can also pick up carbon monoxide. So depending upon the time of year, as the fall moves on and you're moving into fall and eventually winter, and especially in the winter time and a little bit into springtime, you have your furnace on. And so the furnace itself generates carbon monoxide as it burns the fuel. And if that carbon monoxide isn't properly vented out of the house, or even from a car or something like that running, you go out to the garage and you start your car running, that can build up carbon monoxide in the air. It's colorless, it's odorless, we can't see it. And the downside is that carbon monoxide will go and bind over to our hemoglobin. And it binds really strongly to it, so it really likes it, which makes it very difficult to get it off if that should happen to occur. So our red blood cell is really an amazing structure that we have. And so in the smallest amount of blood here, so in a little drop of blood, we've got as many as 5 billion red blood cells. Inside each one of those red blood cells, inside of each red blood cells, we've got some 250 million hemoglobin. So 250 million hemoglobin times 4 oxygens means we're going to have upwards of 1 billion oxygen molecules in just one red blood cell. Multiply that by our red blood cells, and so we now have some 5 billion oxygen molecules needed in order to get all of the red blood cell saturated with oxygen. Normally, we're going to be looking for oxygen saturation to be above 95% on there. And so we're going to look for that to occur. Once we drop below 94%, we start to, on just regular air, typically this is where we're going to start to see that a person may need some supplemental air on there in terms of oxygen. and so they may need to go on a nasal cannula. If it can't be held up there, then we start to look at something like a BiPAP, and then even to intubate them if they still can't keep their levels up well enough either. So erythrocytes themselves, their numbers that are there in terms of the red blood cells and the need that's there basically comes down to monitoring the amount of oxygen that's being carried in the blood. And if our oxygen levels are constantly low, then those oxygen levels are going to stimulate the release of erythropoietin. And so our kidneys are going to say, hey, we need more oxygen in the body. We're in a hypoxic event kind of chronically. And so we're going to need to make some more red blood cells. And so it releases the hormone erythropoietin, and that's going to go off to the red bone marrow. And then the red bone marrow is going to say, okay, we need to make some more red blood cells. And so from that lack of oxygen in the blood, essentially, in a chronic manner, we'll then have the release of that erythropoietin to get us some more red blood cells. And so whether that's because we have decreased numbers of red blood cells, and so we may have lost some blood, so say you go and you donate some blood, all of a sudden now your ability to carry oxygen in those red blood cells is decreased because we just don't have as many. So the kidney says, all right, let me release some erythropoietin. That erythropoietin travels through the bloodstream, heads over to the bone marrow, and the bone marrow starts to produce some more red blood cells. More red blood cells means more red blood cells in the blood, means an increase in the carrying capacity of the blood cells themselves, and we get back to a point where we now have normal red blood cells. And so we have a normal amount there. If it was because of an anemia, person is deficient in a nutrient vital to red blood cell production, no matter how much, in a sense, erythropoietin the kidneys produce, if we don't have all of the products needed or all of the components needed in order to make those red blood cells, we're not going to get normal red blood cells. And so that's going to lead us into an anemic state where we need to then correct whatever it is that's making it so that our red blood cells are normal, correct that, and then we can get some red blood cells that will help out in that process. The other main cell that is produced is the white blood cell. And so our leukocytes, or white blood cells, these are the cells that are going to be our immune cells. And so they're going to go after bacteria, viruses, parasites, things that aren't supposed to be in the body, things like tumor or cancer cells. They're going to clean up after we've had damage somewhere. So all of the debris and dead cells. So you go out and you say, burn yourself or sprain your ankle, something like that. We've damaged cells, and that creates kind of dead cells and debris from the tissue and things like that. And those white blood cells are designed to be able to come around and clean those up. Unlike red blood cells, where we only have one cell type, in white blood cells we have multiple. And so we have many different kinds of the white blood cells. And so as we look at the white blood cells, to remember the different kinds, we have mnemonics that we can use. Never let my engine blow. So the first letter, neutrophils, lymphocytes, monocytes, eosinophils, and basophils. So never let my engine blow. And on the next one, we'll kind of finish that mnemonic, the saying there, with the kind of general idea of the numbers of those cells that we should have. So we have different kinds because they do different jobs. And so things like neutrophils, kind of our neutrophils and our monocytes and our lymphocytes are going to be kind of our general infection types of things as far as what they're elevated for and why we need them. And so neutrophils themselves become phagocytic, so they're going to consume the actual thing that is being invaded or that is invading, whether it be a bacteria or a virus or a viral-infected cell. They're going to become elevated and destroy them by consuming them. Eosinophils we don't really see too many of them because one of their main jobs is when we have parasites. And so most people don't have parasites most of the time. So we're not really going to see a whole lot of eosinophils, but eosinophils can also be utilized to kind of clean up after the basophils. And so the basophils, we're going to see them releasing things like histamine. And when histamine gets released, this is typically when we have some sort of inflammatory condition. And so basophils we're going to see when we have things like allergies. We're going to see them when we have kind of things like the cold. And so that inflammatory process that's kind of going on inside your nose and you're getting all stuffy and everything, whether that's from allergies or whether that's from an infection, that's typically coming from basophils releasing histamine on there. So if we've got a whole bunch of histamine, the eosinophils can come along and clean it up afterwards. They can get rid of it for us. Monocytes are the largest of our white blood cells. They're very big cells. They are big macrophages. They're going to go out and again kind of clean things up. And so they're going to go out and destroy things that are not supposed to be there, things that are infected cells, and get them out of the body. We talked a little bit about lymphocytes for the lymphatic system. And so we have B lymphocytes and we have T lymphocytes. They get their names based upon where they have been matured. And so B lymphocytes come from the bone marrow, and T lymphocytes are coming from the thymus as far as where they were matured. So B lymphocytes, as soon as they leave the bone marrow, they're ready to go. They're ready to destroy whatever it is that they should happen to come in contact with and make antibodies in order to do that. For T lymphocytes, they need to go and spend a little time in the thymus after they've been released from the bone marrow. So they're going to have to go out to the thymus. They become matured there. They become determined that they're immunocompetent, and they can go on their way from there. So the big difference between B lymphocytes and T lymphocytes is that B lymphocytes produce an antibody. They don't actually go out and actively destroy their antigen. T lymphocytes do, though. They're directly destroying cells. And so they're going out and they're kind of actively fighting the fight. B lymphocytes are kind of like the helpers in a sense. Although they're not helper cells, we have those as well. B lymphocytes are there for assisting in the process. So they're going to make these antibodies. Those antibodies are going to create an antibody-antigen complex, and they're going to kind of bind them all up and allow them to be destroyed by our other white blood cells. The last of our formed elements are the platelets. And so the platelets are not a whole cell. They are simply a group of pieces of cells that we have there. And so in this particular picture, we just have red blood cells and some platelets that we can see there. They are, again, coming from stem cells inside the bone marrow. So they're coming from those cells. And so these megakaryocytes that they arise from, they simply kind of give off little pieces of them. So if we have a whole cell here, in terms of our megakaryocyte, they're just going to kind of give off little chunks of themselves. And these little pieces are going to come out of the cells, and those are going to go out, and they're going to serve as the plug, in a sense, during blood clotting. And we'll talk about that in the next video as we start to talk about the hemostasis process, the blood clotting process, and where platelets fill their role in that in terms of generating a platelet plug. And so we'll talk about that next time.